In this video, we're going to discuss sugars. Sugars are a vital component of life. They are used not just for energy or to make things sweet, but they are structural components. They are signaling components for identifying your cells to your immune system or which cells or which parts of your body. They are vital for many, many purposes, far more than we generally give them credit for. They are also very complex stereochemical compounds. So we're going to start with some terms for sugars. Usually this term is carbohydrate. Sugars are carbons with lots of H and OH attached. As a result, they kind of look like carbon with water attached. The name actually comes from hydrated carbon, which is what they originally were thought to be until we discovered their structures. There's a lot of terms for them, however. In this case, often we will hear things like Saccharides. And yes, there are two C's. Saccharin, kind of sweet things, overly sweet. Saccharides are sugars. Well, there are types of the saccharides, and so there are things like polysaccharides, which are linked sugars. Sugars often form chains linking many, many individual sugars together into very large biomolecules. You can have monosaccharides, which are single sugars. You can have disaccharides, which is two. You can have oligio, which is about 3 to 10. And then you can have poly, which is greater than 10. Monosaccharides tend to be used for energy and things. Disaccharides tend to be common ways of obtaining sugars. Or sucrose, table sugar, is also a disaccharide. It is made out of a glucose and a fructose, each of which is a monosaccharide that has been stuck together. And so sugars come in a large variety of individuals and then larger complex mixes as well. So what is a sugar? What makes up an actual sugar in terms of structure? Well, there are two types. There are the aldose and the ketose. Ose is our sugar suffix. Aldose is an aldehyde sugar. The general structure we draw for sugars is to have a carbon with an aldehyde, and then we draw them vertically, these hanging icicle structures. And on these we add the atoms. Sugars have an OH and a hydrogen on each of these carbons. And which side the OH is on varies. What we'll notice is that, well, the aldehyde has 3 things on the carbon. The second carbon, so 1, 2, 3, 4, 5, that second carbon has 4 unique things attached for very different things. That makes that carbon chiral. Sugars have an enormous amount of chirality. Carbon 3 is going to see 4 different things. It's going to see an OH, it's going to see a hydrogen, it's going to see the whole top thing and the whole bottom thing. The top thing has an aldehyde, the bottom does not. They are definitely different groups. Carbon 4 we're going to find is chiral. Carbon 5 has 2 hydrogens. They're identical. The last carbon in the line isn't chiral. But this has 3 chiral centers, which means you could have the OH on the right or the left of your first one, so 2 options. Same with your second OH, same with your third, so 2 times 2 times 2. There's 8 possible configurations of right and left direction connections. This 5-carbon aldehyde sugar has 8 different versions that can be created. In terms of sugars, we don't name every single set of those directions, but we do have names, and they are unique names for some of them. We will not have to learn those unique names for this course. That is not part of our section. We will learn how to recognize the difference, though. If we kind of call one side A and one side B, this has a pattern that says A, B. Equivalent would have been B, B, A. We don't care which side for figuring out this pattern. We just want to know that, hey, my first chiral center went one direction. My second, was it the same or was it different? My third, was it the same or was it different? And what we find is that, oh yeah, our first one is whichever side, our second is the same side, our third is the opposite side. So this is start, same, opposite. If you make the mirror image molecule, so aldehyde, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So let's flip all of those chiral groups. H, OH, OH, hydrogen, hydrogen, OH, hydrogen, and it doesn't matter on the last one because it's not chiral. Well, if we look at which direction our OHs go here, we have the same idea. It starts on one side, goes to the same side, and goes to the opposite side. So regardless of left or right, we can still consider this in AAB type pattern. That pattern, start on a side, same side, opposite side, that pattern has a name. And so what we actually do is we name based on those patterns. This would actually have exactly the same name. This pattern on a 5-carbon aldose, so this is a pentose aldose. So we actually say the number of carbons in our sugar. So pent for 5, it is a 5-carbon sugar that is an aldehyde sugar. This pattern is the pattern for talose. Both of these are talose. So we give the mirror image the exact same name, but we still need to be able to tell you which of the two mirrors you have. Chirality is important. If we use the wrong one, it could be dangerous, or at least it won't do what it's supposed to do. So we need a way to differentiate these two. When it comes to naming, we actually look at the last chiral carbons OH. That is carbon 4 for talose. One of them, the OH is on the right, and one of them, the OH is on the left. If your last chiral OH is on the right, so this one on right, then I am a D-talose. If your last chiral OH, or chiral carbons OH, is on the left, then it is an L-talose. D and L is how we can tell which version of a sugar we have. So while that pattern, same side, opposite side, on a 5-carbon aldehyde sugar will always be named talose, whether it is D or L-talose depends on how we drew it. Was that last chiral OH on the right or on the left? When we have larger molecules, by looking at this, it's got 6 carbons, so it's going to be an hexose. It's an aldehyde sugar, so it is an aldose. It starts on one side, and so it starts same, opposite, same. So AABA. This pattern will always have the same name. This pattern is glucose. Glucose is actually a very important sugar that we will use a lot. So remember this: right, right, left, right. Because this is D-glucose. That last chiral OH is on the right. And we can fill in the hydrogens. Sugars will always have an OH on one side, a hydrogen on the other of those chiral carbons. Often we'll leave the hydrogen off just for clarity, but I'll put it back in here. If we look and try and find chiral centers, there's 4 things, there's 4 things, there's 4 things, there's 4 things. The bottom one again has 2 hydrogens, so it's not chiral. The last chiral carbon is the 5th carbon, and its OH is on the right. So that is a D-glucose. If you've ever brewed or used a few things, you might have seen it called dextrose, which is just a special extra name for D-glucose because we use it so often in humanity. It is the primary sugar that we think of when we think of food and energy metabolism. What this lets us see is a few components. So to tie a few components up for more naming, you have things like triose, tetrose, pentose, hexose, you can heptose. Just how many carbons long is the sugar, so they can be different sizes. You have aldoses, and you have ketoses, depending if it's an aldehyde or a ketone sugar. And a ketone sugar would just have a ketone at the top instead of an aldehyde, which means you have to have 1 carbon down. So here's an example of a pentose ketose. In regard to terms, we have some names for sugars that we will see. We will dive into connections and linked sugars later. But one last bit about our drawings. Our sugars always have our functional group near the top. Whether that's an aldehyde or a ketone, we dangle them down in these chains. We put the functional group as far up as we can, and then we put in all the OHs. This is a Fischer drawing. And it's critical because Fischer drawings allow us to see the chirality on each of these carbons. Because while we draw it sideways, this is really coming out at us. These molecules have been twisted so that the hydrogen and the OH are always coming towards you. So if you hold up one of your hands, twist it so your thumb and your pointer finger are directly towards you, like have them almost touch your eyes, pull them away a little bit. Your thumb is the OH, the finger's the hydrogen, whichever way you want to do it, but they're coming towards you. Whenever we draw a Fischer drawing, this is how the representation goes. Because for any carbon, if you have 2 groups coming towards you, the other 2 groups are going away from you, which means it's possible to determine the left and right-handedness. Fischer drawings are always done this way so that you can be certain whether your OH is supposed to be on the left or on the right. If you have a zigzag drawing, it's a lot harder to identify which side something is on. You don't know if that OH is towards you or away from you. You can draw it in, you can put a wedge, and you can put a dash, but if you hold those thumbs back towards your eyes, well, I have my left hand with my thumb near my left eye and my index finger towards my right eye. If I just rotate my hand around, now 180 degrees, my thumb is now on the right side and my pointer finger is on the left. I can rotate them back. Thumb is on the left side near my left eye. Rotate it away. The thumb is on the right side. As you rotate something, so if we have OH here and hydrogen here, if I spin this around, my carbon is now going to have other things coming up at us. My OH is going to go away. My hydrogen is going to go away. Depending which side you look at, go back to your hand. If it's the side when it's pointed away at you or towards you, it changes the right and left apparent direction of the OH. But you didn't detach your thumb and replace it like you would have to do with atoms on a molecule. You just looked at it from the other side. To make sure we are always keeping our OHs on the correct side, the Fischer drawing is always drawn with them coming towards you. If they could be away, you might accidentally have to flip your molecule around. So this is the idea of a Fischer drawing. We use them to help us keep track of the extensive chirality that occurs in sugars.